I'll kick off my introduction, and I think any other people will be joining in probably rapidly as I'm speaking. But we're really delighted today to welcome speaker Omer Bayraktar. He's been group leader in the cellular genetics at the Wellcome Sanger Institute since 2018. There, his group has been focusing on uncovering the really vast cellular diversity in the human brain using large-scale spatial transcriptomics, imaging, and as well as functional screens. Prior to that, Omer did his postdoc with David Rowich at the University of California in San Francisco and moved with him to the University of Cambridge as a Life Science Research Foundation Fellow. During his time, he developed a large-scale spatial transcriptomics pipeline that's revealed the heterogeneity of astroglial cells across the layers of mammalian cerebral cortex. Prior to that, he did a PhD with Chris Doe at Howard Hughes Medical Institute on specification of neural diversity using Drosophila. Today, though, he's going to be talking about exploring human tissue architecture using high-throughput spatial genomics. It's really great to have you here, Omer, and we're really excited to hear your talk. So I'll hand over to you.
Thank you so much for that introduction, and it's a pleasure to join you all today. So as Rachel gave in that, in that introduction, today I'll tell you about our work at the Sanger Institute on understanding human tissue architecture using high-throughput genomic technologies that allow us to basically do sequencing of cells within tissues. So I wanted to start out with actually my biological point of interest rather than just sort of like my technical occupations. So I'm a neurobiologist by training and trade. And a lot of the questions that I've focused on throughout my training and still wrestle with in my lab involve the significant heterogeneity of cell types in the nervous system. So to the uninitiated, there's a significant diversity of neuronal and non-neuronal cells in our nervous systems that basically support and provide neural function. So neurons themselves come in this really dizzying array of different shapes, molecular properties and functions. And, for the neurons to do their jobs correctly, lots of different non-neuronal cells, the glial cells, consisting mainly of astrocytes, microglia, oligodendrocytes, and vascular cells, also need to support them. And we now know both in the healthy and the diseased nervous system, basically, the roles of each of these different cell types are really critical, and the different conversations that they have with each other here diagrammed in a grossly simplified manner are really important to both the development and the function of the healthy nervous system, but also disease, right? So, I'm fascinated by the cellular complexity, and one of my goals is to really be able to sort of decipher all these conversations that these different neural cell types are having with each other in high throughput. While again, I find this complexity really, a really attractive thing to study, sometimes I'm also really depressed by it. Just the sheer, number of cell types that are involved, the sheer number of different cellular processes to the development or pathology of the nervous system involved, and the vast amount of molecular underpinnings that we are still yet to discover to really understand these processes. And at the Sanger Institute now, my work is really focused on developing high throughput technologies that would allow us to study the cellular architecture of complex tissues like the brain at scale in as few shots as possible. And the remedy that we provide to this complexity is basically multimodal genomics. The workflow that I'll present to you today, starts with basically an unbiased technique that allows us to sort of like map out the diversity of different cell types and their transcriptomes. So that is suspension single cell omics. The idea here, as most of you I assume are already familiar with, is to start with a chunk of tissue, blended it into a soup of single cells, then assay the transcriptome, the chromatin accessibility, and increasingly many other omes of single cells in high throughput. When you do this, you can end up with an unbiased or sort of half supervised classification of different flavors of cells, and for each cell type you have their sort of molecular information, their transcriptome, their chromatin accessibility, genetic regulation in hand, so you can use you know, types of are, you know, thousands to millions of cells per experiment, and it's really easy to do this type of stuff. However, to really start thinking about how all these different cell types in the brain or other, solid organs are communicating with each other, all these cell types are actually living in very specific microenvironments in the tissues, where they're at a particular location in the tissue. They reside in a, very sort of a tailored tissue microenvironment where they have certain, very specific neighbors that they exchange signals and have conversations with. And what suspensional single cell omics doesn't do is that we don't really get to capture this spatial information. So to give us that level of information, the second thing that we do is to, perform spatially resolved transcriptomics and other omics to be able to take these cell types and map them into the 2- and 3-dimensional architecture of the tissues. So with this in hand, now we know where each of these cell types are in 2D and 3D, and we also have access to their transcriptomes. Then the final sort of step of our protocol is to now look into modeling, the interactions between these different neighboring cell types and link them to different phenotypes and, molecular outcomes. So the example that I provided here is to think about, say, 2 neighboring cell types that are expressing complementary, receptors and ligands that can allow us to postulate that they are exchanging signals with each other, and then thinking about downstream sort of transcriptional effectors of that process that might be linked to, sort of, for example, as shown here, sulfate specification or plasticity. So I know that a lot of you are, experts in these different fields and they're interested in different ones. Today, I'll be focusing mainly on how we are utilizing, the spatial transcriptomics part of this, because I think that is sort of one of the newest technologies, in this space. And I'll also try to give you a couple examples of how we are thinking about applying this multimodal genomic workflow in spatial genomics to questions of human disease. So with this combination of single cell and spatial genomics, the way I see it is that it's really, 2 technologies that can allow us to map tissue architecture in high throughput in whatever context you prefer for a solid organ. The layout, the sort of like approach, or the game plan that you saw in the previous slide is more or less, I think the idea here. A lot of the workflows we do, we start with single cell RNA sequencing, which gives us that, single cell resolution, high throughput, and access to, tens of thousands of genes expressed in cells. So, that transcriptome-wide information. After that single cell RNA-seq, we perform various types of spatial genomic approaches that come both in sequencing and imaging-based flavors that provide us different advantages. And what we would like to accomplish is to really put these different data types together to come up with a multimodal representations of the tissues. So we can map all the cells to where they belong in the 2- and 3-dimensional architecture of the tissues, but we also have access to their single cell omes that can allow us to do, high throughput, genomic data analysis. And we're quite excited about, applying this not just to healthy tissues as part of our work in the Human Cell Atlas, but to lots of diseases of the nervous system, as well as other organs and diseases as well. So we are quite interested in sort of pretty much all corners of this diagram here, both from an experimental and computational point of view. But today I'll be focusing mainly on how we are putting the sort of 2 unbiased high throughput technologies, single cell and spatially resolved sequencing, to come up with multimodal, comprehensive tissue maps. So spatially resolved sequencing might sound like a mouthful, but it's actually, simply taking a thin slice of tissue, positionally capturing the transcriptomes of cells from that thin tissue section, and then, reading them out with next generation sequencing. There are a couple of different protocols, but again, the main approach is more or less similar. You start with a thin histology-like tissue section. This used to be mostly, most assays required snap frozen tissue, but we are also now stepping into the age of technologies that can work with, fixed, paraffin embedded archival material as well. These thin tissue sections are generally 1 to 2 cell layers thick. And the idea is that these are placed on various type of arrays, microarrays for some protocols, beta arrays for others, where, you basically have these positionally, placed RNA capture oligonucleotides. Each oligonucleotide has a bit that can capture mRNAs, but it also has a position-specific identity code, a sequencing code. And throughout these protocols, we basically permeabilize the tissue section, so all the mRNAs snake out. They're captured onto the arrays, and through just simple molecular biology, they are stamped with their positional, identification code, and then read out using sequencing. So to the genomicist, this is quite attractive because it gives us unbiased transcriptome-wide analysis with the power of sequencing. The protocols themselves are, based on rather standardized histology and commercialized molecular biology kits. And more importantly, perhaps, for some of the sort of fine tissue mapping work, the spatial resolution of these techniques have been rapidly improving. So, when these technologies were invented several years back, the sort of first incarnation of spatial transcriptomics, the sort of smallest spatial unit you had on these microarrays was about 100 microns. So, that corresponded to many cells. And there are big gaps near adjacent spots, so you were getting rather incomplete profiling of the tissues. The current state-of-the-art commercialized technique, the Visium spatial transcriptomics method from 10x Genomics, the spot size is now down to 50 microns, so that corresponds to 2 to 5 cells on most tissues, so that's much better. And the gaps between adjacent locations is small. And in the sort of space of both homebrew methods as well as updates to the Visium pipeline on the way for next year, we're looking at now significantly higher, resolutions from 4 to, some methods even give you nanometer resolution, several hundred nanometer resolution. But for example, the new Visium method will give you that 4 micron resolution. We are looking at really almost single cell subcellular level profiling of these tissue sections. So we're quite excited about these technologies, but one thing that we've acknowledged from the beginning is that the data that you get out from them is not necessarily the same as single cell RNA-seq. And the simple reason for that is that none of these methods, high or low, preferentially profile single cells. With methods like Visium, it's obvious that each location is capturing multiple cells. So, the information you get from there is like basically the bulk profile of multiple cells, will include multiple different cell types. But even with high resolution methods like this, what you're doing here is that you have a basically regular grid. And when you put a tissue section on there, cells and tissues are not organized into regular grids. So you seldom get a one-to-one correspondence between a cell and one of these array positions. Often what happens with methods like slide-seq is that each position profiles fractions of multiple cells. So both across all of these basically spatial sequencing methods, if you want to get cell type specific information, if you want to be able to specifically say this particular cell type is there versus that other cell type is in this other tissue location, you need to actually computationally deconvolve the cell type specific information. And that is what my lab set out to do when we started at the Sanger Institute. The computational method that we put together as part of a fantastic collaboration with Oli Stegel and DFKZ and EMBL is called saltolocation. So this is the brainchild of a graduate student in the lab, Vitaly Kleshchevnikov. And the idea here is to basically put single-cell RNA-seq and spatial transcriptomics together in a probabilistic manner to deconvolve cell types in spatial transcriptomics data. The simplified protocol is shown here. What saltolocation is doing is that in a sort of a Bayesian manner, we are using single-cell RNA-seq as a cell type dictionary to define the gene expression signatures of all the different cell types in our tissue. And then we are deconvolving our spatial transcriptomic data to be able to map the locations, to estimate the locations of every single cell type in the reference in one step. And with this information in hand, which could amount to the spatial positions of dozens to hundreds of different cells, we can really start asking higher order questions about tissue architecture. We can look at identifying tissue regions with different cell compositions in an automated manner, but in a sort of also in a more exciting manner, we could start looking into now these questions of cell interactions. What cell types are more frequently next to each other than by chance? And are there any sort of receptor ligand expression patterns associated with them? Are there downstream transcriptional, effects of that? So I'll just give you a very, brief, look sort of into what cell location does. So in our protocol, we start with a basically single cell RNA-seq profile. This could be different technologies. This could be cells or nuclei. What we do is basically we extract the average gene expression any of these different data types. And then the algorithm is basically a probabilistic estimation of, the mRNA contribution of every single cell type in the single cell reference to every single spatial position in the spatial transcriptomic data sets. And obviously the advantage of this approach is that instead of just looking at 1 or 2 cell types at once, if your single cell RNA-seq reference has 60 cell types, 100 cell types, all of them are mapped to their tissue locations at once. So to sort of demonstrate to you that cell location works, I'll start telling you about this first pilot test drive data set that we generated in the mouse brain. I assume many of you are very familiar with it, but the mouse brain is a fantastic model system to study cellular heterogeneity because it already has a very well documented sort of rich list of different neuronal and glial cell types. We know very much about the anatomy of the tissue itself, so we know that different mouse brain regions have a different molecular complement of their cell types. So it's a really fantastic paradigm, a breadboard for regional diversity. And what you're looking on the left here is a single nuclei RNA sequencing data set of the mouse brain that we generated, mapping roughly to this brain region or anatomical level that constitutes multiple brain regions. The data set was generated in an unbiased way, so we have lots of different neuronal and glial cell types in it. And by doing a supervised annotation here for standard single cell protocols and known markers of cell types and prior knowledge, we could identify 60 neural cell types, that represent a rich amount of diversity. On the right, I'll play you a little animation that will show cell location in action. So at the beginning of this animation, you'll see our single nuclei reference. I'll have, I've marked here 6 different cell types in different colors. These cell types are, as some of you may be able to read from these labels, are different known neuroglial cell types that we expect to see in different parts of the brain. Throughout the little animation, you'll see how cell location, calculates their average gene expression profile. And then you'll see a histology image of the mouse brain section profiled with the Visium assay. And then every single spot here is basically like one of these Visium spots profiled by the assay. And the different colors demonstrate to you the estimated abundance of these different cell types in different regions. So I'll play that one more time. We have our cell types identified from the single nuclei data. We have our Visium data sets, and you can see these cell types basically map to different brain regions, ranging from different layers of the cerebral cortex, as well as different subcortical structures. Luckily for us, the patterns that we identified here, the spatial locations of these molecular cell types nicely match what we already know. So that gives us lots of basically, confidence in that this method is working. But however, what we really wanted to do with cell location is high throughput mapping of all cell types in our reference, not just sort of 6 cherry-picked cell types. So once we know this is working, we could actually move to the next step, which is taking all of the cell types, looking at all of that data. And the way we do that is largely automated protocol, where on the left here, you can see the anatomical, sort of the histology image of the tissue section profiled with the Visium assay. In the middle, what we have done is that we have taken every single Visium spot, that's 55 micron Visium spots, and we have simply clustered them based on the estimated composition of every single cell type by cell location, single cell reference. That has provided us basically the spatial regions that very nicely correspond to the anatomical compartments of the brain, cortical layers, subcortical regions, hippocampal fields, thalamic nuclei, etc. And with these in hand, basically what you can start to build is this really high throughput map of this tissue. So we have, 29 regions identified by the cell composition clustering, and then we have 59 cell types coming from our single cell data deconvolved in the spatial transcriptomic data set across locations, cell types, and across many, but also size to very specific areas. We have a couple of really nice examples here, such as fine regional astrocyte subtypes that we covered that were not previously on the mouse brain. I'd love to tell you more about those if you're interested. Use this technology and this sort of multimodal approach to take lots of cell types and in one shot map their spatial positions in the tissue. So the mouse brain work was really encouraging to us, but we know that, the mouse brain is a fantastic system for regional heterogeneity. It doesn't represent the spatial architecture of many other human tissues. Lots of human tissues, such as immune organs like the lymph nodes, instead of sort of these very regionally compartmentalized cell types, actually have slightly more complex structures where they can have, closely interlinked immune cell states that are actually sort of spatially interlaced, found out through spatial gradients in the tissue rather than discrete regions. So we are quite interested to test drive cell to location, in a context like this. And to do this, we did what you're seeing here on the slide, where we put together, again, a single cell reference of the human secondary lymphoid organs that give really fine, good representation of different immune cell types, as well as, stromal cells. Then we took a Visium data set of a human lymph node, and when we performed, cell to location based mapping of this reference, we could nicely see these cardinal, basically, compartments of the lymph node presented by major cell types. These include the T cell zones, the B follicles, as well as the germinal centers. And what we wanted to do after this, was to really see, again, can we repeat what we did in the mouse brain? Can we arrive at a higher order representation of tissue architecture? And the way we did that was slightly different from tissue region identification. What I'm showing you here is now a second layer of deconvolution of cell to location results, where we took all the cell type mappings from cell to location and then used non-negative matrix factorization to be able to ask, what are the cell types that are actually co-located with each other? And what this reveals are basically these grouping of cell types that nicely correspond to known and novel, basically, groupings and nodes. The two that highlighted, where sort of you can see, these different cell types that are identified to these different sort of groupings or factors that show spatial profiles that allow us correspond to different of T and B cell maturation, basically these cell states in the tissue microenvironments. So this really gives us a fantastic, I think, place to start doing that interaction analyses, now knowing that these two cell types are co-located with each other versus these four cell types that preferentially travel with each other. And finally, before I move on to the disease applications, I also want to show you a little bit of data on how self-location is different than some of the other sort of spatial transcriptomics deconvolution methods out there. So those of you who have their fingers on the pulse of this technology are probably aware that last year saw lots of fantastic computational methods being published. And I think a lot of them have really interesting and complementary strengths. But one thing that's we have found that cell to location excels at by design is the ability to do detailed mapping of fine-grained cell types in complex tissues. And what I mean by that are, these fine-grained, transcriptionally similar flavors of cells, such as immune cell types, that are found in many tissues. There are also lots of different examples of these, fine-grained cell types, both in the nervous system as well as other organs. Again, the idea here is that these are cells that are not necessarily showing the broadest of the transcriptional differences, but they're often known to have really different critical functions, and really the ability to be able to resolution to discriminate the locations of these different cell types is really important to do that multimodal genomic mapping I talked about at the beginning. So to do this, we actually turned our attention into another organ. So we looked into the human gut, of which the Sarah Teichmann's lab had generated a single-cell RNA-seq reference for. One of the fantastic things in this study that got published this year was that it had a very detailed annotation of immune cell states, including very fine-grained differences, totaling, I think, something about 77 cell states. And what we could do was sort of repeat that two-step protocol I showed you before. We got then a sort of complementary published Visium data set of the human colon from Alison Simmons's lab in Oxford. And what we could do is that we could basically map the single cell reference and assess if we were successful in identifying these fine-grained immune cell types in the right locations. One of the reasons that we picked this tissue, in addition to the fact that we have this very detailed reference, was that we have a very nice ground truth where immune cell types should be. So if you look into these histology images of the human gut, you see these sort of like dense like nodes here and there. So these are secondary lymphoid follicles of the gut, these tertiary lymphoid structures where we expect to see immune cells. And indeed, when you do cell to location mapping of the major sort of cell types of the gut, ranging from immune cells to colonocytes to epithelial cells to myofibroblast, you see that indeed this is the immune cell type location, and all the other cells are also where they're supposed to be. So with this type of tissue, we have this nice, basically expected cell type location that we can use for a quantitative benchmark. With that in hand, we can actually move into the sort of challenge to cell to location and other methods to whether they could map different subtypes. So what I'm showing you here now is taking the immune cell types in the reference, and I think we picked about 19 of them that had sort of published data on their location to these lymphoid follicles. And here, what we have done was we could basically look at single cell profiles and use quantitative methods to determine if a cell type was very transcriptionally distinct, if it showed lots of differences from other cells, versus whether it was similar to other cells, whether it showed rather fine-grained differences. And in both cases, cell to location could map these both distinct and fine subtypes to these lymphoid structures with high success. And the fine cell types that we are dealing with here are, again, cells that are quite important to tissue function. These T follicular helper cells, for example, are perhaps not as different from other T cells as cycling B cells are, but they are quite integral in guiding B cell differentiation into these lymphoid follicles. However, when we looked at other technologies that were recently published, other deconvolution methods, we found that while they were all very good at mapping transcriptionally distinct cells, they often struggled a lot in locating, fine cell types in immune locations. Supporting our claim that cell to location is overall issue in a more. Again, quantification of the performance of the different methods is highlighted here, where the take home is if all you care about is broad cell types like fibroblast versus immune cell types, any method out there is fine. But if you would really like to ask questions about, where fine cell types are and how they relate to each other, you should use a method like self-allocation. So, since its inception in the lab, it's been really a pleasure to work with a bunch of other groups at the Sanger Institute and elsewhere and apply self-allocation to various different human tissues. Now we're really proud to present this work where we can, demonstrate high throughput mapping of tissue architecture across really different human organs, from the human brain to immune organs to the developing gut. So my lab now is quite interested, as part of our core research portfolio, in taking these technologies we've built and applying them to the diseases, sort of both the healthy and the diseased human nervous system. So, aligned with this, we have basically started generating these multimodal data sets of the human brain. So what I'm showing you here is an unpublished data set from our lab, where we have obtained high quality neurosurgical biopsy samples of the human cortex from adult patients and performed unbiased single nuclei sequencing to identify fine subtypes of neurons and glial cells. And, it's been really helpful to get to use an unbiased technology here and get a good grip on some of the cell types that are often neglected in other studies, chiefly the glial cells. And, we're also quite interested, as part of our current portfolio, in taking all these cell types and mapping them to the Visium data of the adult cortex. What we have done here is to take a published data set of the Visium data set of the adult human brain, and we mapped our reference to it. And those who are familiar with the anatomy would recognize the correct mapping of cortical layers here. So we can start asking these types of high throughput, multimodal genomic questions. So before I start talking about some of our disease, focuses, I would also like to kind of highlight some of the unpublished work we are doing in kind of like the next step of this pipeline. We had single cell data. It gave us a sort of a inventory and transcriptomic profiles of cell types in a tissue. We do our spatial genomic mapping that can locate all those cell types in the tissue space and identify which cell types are co-located. But then it comes the time to start modeling cell-cell interactions. And something that we'd really like to exploit is basically this receptor ligand expression-based analysis. If at the level of both the either or the single cell or the spatial data, if you have these expression of extracellular receptors and ligands that are known to interact with each other in different cells, you can actually postulate or hypothesize their interactions. We would really like to be able to use this information to gain better insights into how different human brain cell types interact with each other. And chiefly, specifically, we're quite interested in how glial cells, microglia and astrocytes interact with each other and neurons in both the healthy brain development and the development of synapses, but also in various neurodegenerative disorders, such as ALS, but also, as you'll see at the final part of my presentation, in brain cancer. However, as we started, looking into this part of the cell interaction mapping in the brain in more detail, we identified a challenge in that a lot of the insight we have, a lot of the sort of curation we have on which receptors bind to which ligands actually comes from the study of the immune system. And if we were to do a quick comparison based on single cell data of sort of multiple human brain data sets compared to multiple human immune cell data sets, and we identify all the sort of extracellular receptors and ligands in these data sets, and cross-reference them across databases to see which ones are actually annotated to have interactors, we see that the neural receptor ligands are generally poorly mapped out. So this diagram here shows you the frequency of sort of the brain versus immune, receptors. And you basically find that in the brain, compared to the immune system, there are more orphan receptors that do not have known binding partners. And those with known binding partners often have many fewer, partners than immune cell types. So there's basically a lack of knowledge, high quality information on which brain receptors are binding, are interacting with other brain receptors and ligands. So as part of the collaboration we had at the Sanger Institute with Gavin Wright, who's now moved his lab to the University of York, we were actually quite interested to close this knowledge gap by performing high throughput biochemical surface receptor interaction screens. The method that I'll cover today in the next few slides, it's called Savexis. It was developed by a fantastic grad student in Gavin's lab, Jared Schultz. And while I don't have a lot of time to cover the details, the idea here is to basically, use high throughput recombinant protein production and, coupled with, biochemical one-on-one protein interaction screening to be able to build physical interaction maps of hundreds of proteins in a scalable manner. With these protocols, again, we start with the sort of a knowledge of, two proteins, say, cell type A and cell type B expressing protein A and protein B. We basically clone the ectodomains, the extracellular protein using use 384 value. Basically at the bottom of streptavidin encoded plates, and then we add the prey proteins coupled to HRP tags that chromogenically, if there is binding, basically, the sort of simple visual and spectroscopic, quantification of a 384 valve plate like this shows you an interaction by color. And for us, the advantage here is that this is really something you can expand to hundreds of proteins in a matter of several weeks to a few months. So it really allows us to close this gap of brain receptor interactions in a feasible manner. So what we have done here now with Gavin's lab is we have completed actually an initial screen where we looked at single nuclei RNA sequencing, our single nuclei RNA sequencing data set of the human brain. We focused on astrocyte microglia neurons. We pulled about 512 single transmembrane surface receptors and ligands from these cell types. And we basically screened their one-on-one interactions of all these 500 proteins, adding up to almost 200,000 interactions screened. And the map here is basically, protein 1 versus protein 2 type of quantification. So this massive heat map is really demonstrating to you the signals that we have identified. So we are still in the sort of, biological interpretation phase of this project. So what I'll share with you for now is that when we actually look into the interactions we have mapped out, there's very nice concordance, there's very nice consistency between our results and the high quality interactions of the human brain cell types, human brain receptors that are already known. So this little map here again sort of shows about, I think, 40 pairs of interactions that we pulled. These are very high confidence based on our method. And when we look into these protein interaction databases, there's also very good biochemical evidence on the interaction of these guys. And you can see lots of sort of interesting and biologically relevant targets here, from axon guidance molecules to, the MERTK pathway, which is responsible for, synaptic engulfment in the brain, as well as other proteins. So we're quite excited here to dig deeper into the screen results and identify, and we have indeed identified actually a bunch of novel interactions that I can't share today, but we're quite interested into diving into rare biology and enriching sort of the, our knowledge of cell interactions for this high throughput multimodal analysis. So as I'll close, I'd like to tell you a little bit about what human diseases we're actually applying these multimodal genomic technologies to. I talked about this elsewhere, so I'll just skip this with a slide, but I just wanted to show that, what was sort of, like one obviously disease that we wanted to help out with if possible was COVID-19. And as part of this work, as part of a multi-center collaboration between the Sanger Institute, Imperial College, University of York, and the University of Newcastle, funded by the UK Coronavirus Immunology Consortium, we were able to perform this multimodal spatial genomic mapping of the lungs of patients who passed from severe COVID. And again, the idea here is that we were able to generate these both by our own resources, but also with computational integration with existing data sets, these high quality single cell atlases of healthy and COVID lungs. We could generate spatial transcriptomic profiling of pathologically relevant areas from different lung regions using a different nanostring technology, and we could put these together using a cell to location like approach, which could allow us to really identify aberrations in cell composition, but also the knowledge of which cell types are located with each other, which cell types are spatially traveling with these pathologies, so that they can allow us to dissect COVID-19 pathology later. The disease that I'll focus in a bit more detail now over the next 5 to 7 minutes is actually brain cancer. Specifically, what we've been focusing our efforts on over the lab now is to try and see if we can start looking into the cellular basis of glioblastoma, the cellular heterogeneity of glioblastoma in greater detail using our multimodal genomic approaches. So again, I assume a lot of you are familiar with this, so just a brief introduction here. I think glioblastoma is really a terrible brain cancer. It's a leading cause of life years lost to cancer, with many deaths in the UK. It's uniformly and rapidly fatal, and it's and unfortunately highly insensitive to lots of different therapies. So there's a rather grim outcome for many patients, and there's also significant incidence in younger adults and children. And again, lots of different therapies have now been successful, included immunotherapies. So there are really, I think, there's really a need for trying out new approaches that might lead to new therapeutic insights. I think one of the sort of problems with the glioblastoma is that years of molecular studies, including more recent single cell transcriptomic based work, has taught us that there's a significant amount of dynamic cellular transcriptional and epigenetic heterogeneity. Specifically, several sort of single cell transcriptomics based studies have suggested that these tumors across different patients or across different sort of sites in glioblastoma tumors actually consist of these recurring different flavors of malignant cell types that are corrupt mimics of neurodevelopmental, such as neural progenitors, astrocytes or OPCs, or other cell states that we don't quite understand well yet, mesenchymal cells. And each of these cell states are actually highly plastic, by doing, animal xenograft studies. If you were to sort of isolate one cell state from a patient, put it into an animal, you can actually see other cell states be derived from those cells. And throughout both disease relapse or tumor progression, these cell types or these different glioblastoma cell states are really moving around, zigzagging, and understanding really how all these different cell states are regulated, how they transform from one fate to another is potentially quite valuable to basically short circuit glioblastoma. Maybe we can stop these sort of some of the more malignant cell states, trap the brain cancer in some of these, some of these sort of like more, like less malignant cell states, or we could just use this information to develop genetic approaches to better combat, to better kind of target all these malignant cell states. And one of the factors that sort of come to light over the past couple of years on the regulation of these different cell state of the GBM cell state plasticity and transitions is the tumor microenvironment. There is now, I think, a good deal of evidence that there is this, two-lane communication between the tumor microenvironment cells and malignant cells in glioblastoma, in which the tumor microenvironment primes, some of these cell state transitions, and in return, the malignant cells themselves can actually force the tumor microenvironment, the cells, to support their development, in a way co-opt the host's immune system, for example. So we're quite interested if our multimodal genomic protocol, combining single cell and spatial genomics, could be used to systematically dissect these interactions between the tumor microenvironment and provide us better molecular knowledge on what signals are regulating these malignant cell transitions and what could be some of the gene regulatory mechanisms behind these processes, both at the level of GBM cells, but also the tumor microenvironment cells themselves. So to do this, we were quite fortunate recently to obtain a good deal of funding from the newly established Wellcome Leap Delta Tissue Program. And for this, we put a fantastic team of clinicians, both clinicians, basic researchers, and developmental biologists that are going to work over the next 3 years to use multimodal genomic technologies to generate large-scale data sets and develop new computational approaches to decipher this communication between, malignant cells and the tumor microenvironment. So again, our fantastic team, I think, covers many talents. I'll introduce just a few in the course of this over my next few slides, but we really have a fantastic team. Consists of computational wizards like Oli, really, hardcore neurodevelopmental biologists like James Briscoe, and also, real cancer biologists like San Behchadi, but, fantastic clinician researchers like David Rowich. So, now I think I've shown you this multimodal genomic approach several times, so I won't dwell on it, but our idea here is really, can we profile high quality neurosurgical resection tissue from many samples using these, multiple single cell and spatial genomic technologies that allow us to read now in a joint manner, both the transcriptomes and chromatin accessibility of individual cells from suspension data, the spatially resolved transcriptomes from tissue data, but also spatial whole genome sequencing, which is basically low input DNA sequencing of a few dozens of cells excised with laser capture micro dissection that would allow us to establish clonal lineage than the insight provided by any single one of them. So we're really keen in developing kind of the next generation of cell location and accompanying tools to be able to put together a spatially resolved atlas of GBM that we call GBM space that will allow us to predict these cellular interactions and link them to these GBM cell state transitions. So we have both experimental and computational goals as part of this project, but the only thing that I'll mention today is that we'll be generating, as we've already started, this year, a truly large-scale multimodal genomic data set. So working with a fantastic set of neurosurgeons led by Richard Mayer at Addenbrooke's, we will be profiling at least 50 patients across multiple tumor sites. And through the use of these different technologies, we'll be developing, we'll be accumulating the sort of multi-omic RNA plus chromatin accessibility profiles of several million nuclei, coupled with several hundred Visium tissue sections, giving us good coverage of the different microenvironments, and also a good number of these, spatial whole genome sequencing profiles. We already have, good, I think, pilot data to suggest that even just the RNA profiling alone at the single nuclei level can give us these different cell states. So here, from a small number of tumors, you can see that with unbiased single nucleus sequencing, we could identify malignant cell types in red and all other tumor microenvironment cells. And the malignant cells, we can actually sort them into these different cell states that indeed correspond to what has been published before. They, express gene expression signatures, resembling, neurodevelopmental cell types such as neural precursors or OPCs or mesenchymal cells, as you can see from this grouping. But those sort of hawk-eyed, heat map connoisseurs amongst you might also see that there are actually lots of cells that seem to be hybrid states. There are those, for example, in this OPC-like state that are sort of also expressing the MPC program. So we're quite interested with our expanded data set to dig in further into these sort of intermediate and hybrid states and identify these transitions better. And again, we know with the spatial RNA-seq, our first choice will be Visium. Working with my partner in crime, Sam Bechati at the Sanger Institute, we'll be, starting with Visium, but next year we'll be upgrading to the Visium HD technology, which gives a higher resolution. So we're quite, interested and excited about the, increased resolution this technique will provide to map individual cell types. And also, we're also excited about the challenges with my other computational partner in crime, Oli Stegel, to deal with much larger scale spatial transcriptomic data sets. And again, for spatial DNA sequencing with Sam, we got a fantastic setup at the Sanger Institute where, using laser capture microdissection, we could go into, specific regions or just do a grid type of profiling and basically do low input, high fidelity, or high sensitivity whole genome DNA sequencing that will allow us to establish clonal phylogenetic trees and relate them to these sort of transcriptional changes from tissue sections adjacent to them. But we're also looking to add a bulk RNA-seq to this pipeline as well. So this is my final slide. I hope today, with both the sort of computational method development and biological applications I showed you, I convince you that, we could use these multimodal genealogies now to start doing high throughput mapping of cellular circuits and tissues. And I think what this is really pointing at for lots of human and model organism tissues is really, comprehensive cellular wiring diagrams of, development of healthy adult tissues and disease samples in the future. So we're quite excited about these opportunities. So with that, I'll stop. I'll thank you for your attention. I'll acknowledge my fantastic lab. I'll say that also we are actively recruiting to multiple projects. So if you're interested in any of this, please do get in touch. And thank you.